My name is Anil Dutta. I am a pediatrician by training and currently employed at GSK as the head of clinical and medical for emerging markets vaccines. I'm delighted to share some thoughts with this august audience of dear colleagues today on tuberculosis. I start with a memory I have from the 70s when I was a medical student in India. And we had a great teacher of tuberculosis who would often say two things. One, that diabetes and tuberculosis are two diseases which encompass a broad level of medical knowledge, and that being a student of these two diseases really equips you to understand multiple elements of medical science: radiology, clinical, bedside diagnosis, laboratory diagnosis, treatment, pharmacology. So he would encompass a lot in one sentence. And the same teacher also would say, you are the generation who is going to see TB on decline. So learn fast, because these many patients that you see in my ward today, you will not have that opportunity once you grow up. Like all teachers, he was half right and half wrong. He was right in that tuberculosis is a disease that really develops our medical students' knowledge from a 360 perspective. But he was probably less right when he said that we would see the end of tuberculosis. Of course, those were days when rifampicin was just discovered and was found to be very highly effective against tuberculosis. Those were the days when HIV was not known. So with hindsight, my teacher did have a vision, but that vision, unfortunately, hasn't materialized into reality. Today that we are talking about tuberculosis, we are talking at an era when the disease is rampant. It is in fact one of the largest killers among infectious diseases known to man, and it's been with us for years and decades and centuries. Tuberculosis is an old disease, but on which we keep learning something new every day. In today's talk, I would very quickly gloss over some fundamentals of tuberculosis. I assume this audience is that of experts, and you obviously know the subject much better than I do. I will instead focus on the principles of TB vaccines, and by no means do I know all of them. I'm more conversant with the vaccine we worked on, and I'll just carry us through some of the thoughts I have on the vaccines that are available, on their pluses and their fallacies, on the optimal way forward, and how research in tuberculosis is shaping up in the future. Tuberculosis, once called King's disease, because it was thought that a king's touch would cure the disease. It's a bacterial infection by Mycobacterium tuberculosis, as you all know. It's a disease which is usually very often affecting the lungs. So it's a pulmonary tuberculosis, which is the largest spectrum of tuberculous disease, while it can also have multi-organ manifestations. And in recent past, we have started observing an increasing incidence of drug-resistant tuberculosis. One of the interesting aspects of this disease is, and there are not too many examples in medical science, where the disease can remain latent after a primary infection and then reactivate into an active disease at an older age. Now, there are some other diseases that follow the same mechanism, but tuberculosis has been the first one we really figured out that causes a primary infection, which then can, in some cases, many cases, in fact, lead to a post-primary disease. It can affect other parts of body as well, as I said. So we are aware about multiple forms of tuberculosis, although pulmonary tuberculosis still remains the biggest one. And from a transmission perspective, an open case of pulmonary tuberculosis is still the fundamental center point from which transmission continues to take place. It's a big public health problem. It has been a big public health problem for ages, and it still stays a big public health problem. In 2017, for example, we had about 10 million new cases of tuberculosis. And this is only those which are well captured and notified. The reality may be somewhat larger. We had 1.5 or 6 million deaths from tuberculosis. And these deaths touched the full spectrum. Children, women, men, spares no one. In children, the manifestation of the disease is slightly different because it's a primary complex, a primary disease, and therefore the big killers of children continue to be tubercular meningitis, endobronchial tuberculosis, and miliary tuberculosis. Regarding the treatment, while rifampicin was that magic antibiotic in the 70s when it was introduced, and very rapidly led to a decline in cases which were resistant to INH, ethambutol, PAS, etc. Today we see that more and more cases are becoming resistant to rifampicin, and rifampicin, though being a first-line treatment today alongside other drugs, is not as effective as it was in the 70s, and we are having to go for new generation drugs. So that's another piece of the race that continues. Who among us is at biggest risk? Children are at risk, yes. They develop the primary complex. In a vast majority of children, this primary complex is localized into what we call the Gons focus. So an area of infection in the lungs, which is a zone surrounded by fibrosis, inside of which we find a central zone of caseation. We find some types of giant cells, infiltration with lymphocytes, and the organism itself. Over time, this can either remain calcified as a focus, and then we accidentally discover on adult radiographs the calcification of the focus, or when the conditions are right for the organism, particularly in immune deficient situations, there is a spillover, a flare-up, and then it can either go through the bloodstream or to contagious organs and then lead to a disseminated disease. While TB was a democratic disease impacting all of mankind, and there are multiple stories in Europe on tuberculosis, the King's disease was the example I gave. However, over time, with efficiencies in diagnosis, healthcare systems in general, early access to medical care, availability and affordability of drugs, the disease has become slightly limited to the developing world. And today it is largely present in the developing world, over 95% of cases and deaths. 8 countries among them are the big ones, among which India and China remain the biggest contributors, also due to their population size. What we also learned after the 70s was that the advent of HIV has taught us that HIV positive people are 20 to 30-fold more likely to develop active tuberculosis. There are other risk factors. There is diabetes as a known risk factor, alcohol and tobacco use, malnutrition, other forms of immune deficiency, treatment on steroids, even infections with measles could lead to a post-measles flaring up of tuberculosis. So anything that would impact the immune response, lower the immune response, would be a trigger for a broader disease manifestation. The spread is classical. Initial spread is clearly through the aerosol route. So people with active tuberculosis discharge the bacteria into their air. Therefore, the sputum positive being the biggest diagnostic criteria we were taught. And we remember our teachers telling us finding just one bacteria is worth more than having all the indirect tests and radiographs put together. And that still stands true today. But we do now have other tests like the IGRA, the gamma interferon release assay. These are able to also be differentiators between latent and active tuberculosis. Treatment of tuberculosis, treatment is available. TB is a treatable disease. It's a curable disease. And over time, we can be proud to say that within the last, say, decade, we have saved over 50 million lives with the existing treatments that we have. It's also true that we have lost many more also because of the multi-drug resistance and other factors, or inaccess to treatment or lack of compliance with the treatment. Those are still important factors that we continue facing. The first line treatment like rifampicin, INH, The bacteria that do not respond to this, they cause the multi-drug resistant tuberculosis. And this then requires more extensive chemotherapy with a second line treatment, which obviously increases cost and complexity and availability in some parts of the world. Multi-drug resistant strains have been documented in every country we have surveyed. So we have seen that they are close to about 10% of multi-drug resistant tuberculosis. Now, for some patients, there are really no further treatment options left. The multi-drug resistance is to an extent that nothing else the organism would be sensitive to. In 2014, the WHO launched the End TB strategy. It was a worthy goal. It is a worthy goal. The goal is that by the end of 2030, we should have 90% fewer deaths, 80% fewer cases, and catastrophic costs to families should be reduced altogether. These goals are challenging. They're challenging because the world is a challenging place. Not every situation or environment is the same. However, these are absolutely reachable goals that we must strive for. Because if we, in the 21st century, cannot think of eradicating or not eradicating, but reducing the burden of a disease significantly, which has been with us for centuries, then we have this work ahead of us. This strategy can be underpinned by 3 pillars. So one is patient-centered TB care and prevention. So that's one integrated patient-centered approach. Then policy initiatives and supporting systems that go to reinforce that objective. And finally, the research and innovation needs to continue because despite this being a disease of centuries, we are still learning new things about this. So in 2018, the United Nations held the first high-level meeting on TB, and the world leaders in the General Assembly have reaffirmed their commitment to ending this epidemic by 2030, or at least in significantly reducing the burden of death, disability, sickness, and costs. Research in tuberculosis. Now we come to research and innovation in tuberculosis, and we know that without research, without innovative approaches, with business as usual, we are not going to achieve those 90% goals. So clearly some more information, some more research, some more information is needed, innovation is needed. Better diagnostics, that's an important need. We do have, and we have improved a lot from a time when acid-fast bacillus on a smear was the only way we could look at things, or a poor quality radiograph in the periphery, or a mass miniature radiography that was started. We have come a long way from that time, but that research continues. Treatments need to improve. And there are 2 or 3 ways treatment improvements are being addressed. So one is to reduce the duration of treatment and the complexity of treatment. The old treatment with the para-aminosalicylate, grams of it having to be taken like almost like a snack, that has changed into a minimized tablet form, readily swallowable, direct under observation therapy. All those modalities have been put in place, but there's more work happening there, especially with regards to multi-drug resistance. We have also to remember that we have to be more efficient in our treatment of latent tuberculosis, because 1/3 of these are going to turn active at some stage. So that is another conduit of transmission that needs to be addressed. And finally, we come to the vaccines. Vaccines for tuberculosis have been the first vaccine, and so far the only vaccine in broad use is the BCG vaccine, developed exactly 100 years ago in 1921, which took 13 years and 230 plus subcultures of the Mycobacterium tuberculosis, the bovine strain by Calmet and Guerin. And that is the vaccine which has been the mainstay of tuberculosis. Now, fortunately for us, BCG does induce a powerful immune response, including the cell-mediated immune response, rightly so, because it is at the end of the day, it's the organism itself, attenuated form. But BCG in multiple studies worldwide has shown that it's effective to an extent in children, young children, in preventing the complications of primary tuberculosis. So when it comes to miliary tuberculosis, endobronchial tuberculosis, or tubercular meningitis, yes, BCG is highly effective in reducing the burden there. But beyond that, in the adolescents, in the adults, BCG loses its effectivity, and over time it anyway declines in its efficiency. So BCG alone, while it has been the mainstay so far, is probably not the only way we can achieve this 90+% target that we have in mind. This brings me back to another question, another story about my teacher, the same teacher. He was Professor Goswami, who used to teach us tuberculosis. He would then say, and he was a very strong supporter of the BCG vaccine. And Professor would then tell us that this is a vaccine which, now that we have it, and at that time the problem was in deploying BCG appropriately. He says once the deployment is worldwide and WHO is all behind it, it's a part of the EPI worldwide, very soon we will see that decline happening. He was right in the extent of the pediatric segment. He was not absolutely right in the segment of the older adults that we see today. This particular vaccine, it's a priming vaccine. BCG is a priming vaccine. But then we do not recommend revaccination because there is lack of evidence that revaccination confers additional protection. We have seen in the famous Chingalpet trial from the south of India, where the first time in a large cohort of community study, BCG was found to be underperforming. Today we are in a situation when 1 in 4 human beings are infected with latent tuberculosis. Affected people have about a 10% lifetime risk of reactivation and developing active tuberculosis in future. And the disease in latently infected adults and adolescents today accounts for much of the disease burden and transmission that is happening. So clearly there's an urgent need for a well-tolerated, effective vaccine that addresses or meets that gap which is there. So we have now 2 sets of circumstances in front of us. We have those who are in the pediatric segment, where the primary tuberculosis needs to be taken care of as a prophylactic vaccine. And then we have the segment in which we need a vaccine for those who are in the latent phases and in whom the activation needs to be slowed down or blocked or treated more efficiently. There are thus 4 targets for vaccines being developed. So prevention of the disease is an objective. In children, we have it to some extent, as we just discussed. In adults and adolescents, work is ongoing, and now we have come across vaccines which have proven efficacious to some extent in reducing or in preventing tuberculosis disease in adults and adolescents. We need a replacement for BCG as a BCG booster, so early life immunization, but that's another area of research. Vaccination of TB patients after treatment to prevent the recurrence. That's another important objective. And then, of course, immunotherapeutic adjuncts to drug therapy, which can reduce the duration of therapy. There have been BCG itself has been a powerful adjuvant in reducing or in influencing therapy for other diseases. But for tuberculosis itself, we do need a vaccine which would be able to supplement or augment or partner with the drug treatment to make the treatment more efficient. There are multiple vaccines in clinical trials today, and I have to confess this slide is about 2 years old, so if there is a name or 2 missing, I apologize for that. The type of vaccines that we see today in development, in various phases of development, we have the viral vector approach, so like the Oxford vaccine, the chimp adenovirus approach. We have the protein subunit vaccines, which are among them are adjuvanted vaccines like the M72, and we'll look a little more on that. And then, of course, we have the classical approaches of either a kill or a whole cell or an extract or a live attenuated approach. So multiple studies have been ongoing. Very promising results have been seen in some studies, and I'll be able to comment on the M72 study, M72 vaccine study. which has shown us about a 50% reduction. So these candidates, they have been aimed at addressing current unmet needs. So current unmet needs, as we discussed, was targeting adolescents and adults in the TB endemic region and preventing pulmonary tuberculosis as a need. The vaccine M72 is an adjuvanted vaccine. So there is a fusion protein, the M72, which is actually a recombinant protein taking a full-length MTB39A antigen and flanked by inverted halves of the MTB32A. So the N-terminal and the C-terminal on two sides and the full-length MTB39 in the center, together called the fusion protein M72. This is adjuvanted by the GSK's proprietary adjuvant, which is the AS01E. It's the same adjuvant which contains MPL, which contains QS21. It's a liposomal formulation, and this is a proven adjuvant which has been used for other vaccines as well, including the malaria vaccine. In the clinical development of the M72, so the phase 1 was completed, and this was a study in PPD negative adults, just looking at safety and immunogenicity among them. A phase 2 was completed, which was PPD positive and BCG primed adults. Again, safety and immunogenicity was evaluated. People who are HIV positive or HIV negatives on treatment, their safety and immunogenicity was evaluated, and adolescent safety and immunogenicity, because that was the ultimate end target. Now on this slide, we see phase 2B ongoing. Well, the phase 2B has been completed and published, and we'll look at some of that outcome. This is the phase 2B study, which was conducted in South Africa, Kenya, and Zambia, which were the high endemic regions in Africa. The objective was to evaluate the protective efficacy of this vaccine M72 against pulmonary tuberculosis in healthy HIV negative adults with likely TB infection but without the disease. So they were having likely infected but not having the disease. So they were randomized 1-to-1. One half of the study cohort, which was 3,500 plus people, received M72 in 2 doses, 1 month apart, while the control group had a placebo, which was also given 1 month apart. The primary outcome we were looking for or evaluating there was incidence of cases of definite pulmonary tuberculosis, proven disease, not associated with HIV infection. And obviously, there were other studies that piggybacked on it for the future, which was including the stock, which was created for further research with the consent of the people for their biological samples. Here are the results of the study. So we found about 50% efficacy, which is efficacy being defined, as we all know, as a percentage reduction in the incidence of disease between the vaccinated versus non-vaccinated individuals. So the vaccinated cohort in the previous design that I showed had 10 cases of tuberculosis, while there were 22 cases in the placebo-controlled group, which gives us a 54% reduction. Therefore, that is the vaccine efficacy we talk about. Now, it's interesting here to note that BCG vaccine itself also has about a 50% efficacy, but in those without the active disease. So the side effect profile was acceptable. We had adverse events with the vaccine arm, obviously, because of the content of the vaccine, little higher than the placebo arm, but the serious adverse events were nearly the same, as were potential immune-mediated diseases, as were fatalities. So on all counts, no deaths were considered to be study-related, and the side effect profile was largely comparable with the placebo, barring adverse events which were largely local again. So a clinically acceptable profile. One important question that comes to mind, and these days we hear of COVID vaccines, and we have kind of become used to thinking about a vaccine with 90% efficacy, 95%, and if some vaccine comes with 80%, we are kind of saying this is an underperforming vaccine. It's not so. In fact, 90 and 90 plus efficacies are there for several vaccines. They're there, but they're not the standard norm. DPT doesn't have it. BCG doesn't have it, right? Oral polio vaccine certainly doesn't have it. So it's not so much the number that counts, but it's the impact that counts. For a disease which is rampant and which has a huge burden worldwide of tens of millions of people being infected, a 50% reduction in incidence of that disease with its accompanied impact on deaths and lives saved and its impact on inability to transmit further when put in totality is huge. So this is a game-changing discovery of a 50% reduction in incidence of disease among the adolescents and adults. Where are we heading now? This is an interesting part. So a conscious decision. This was a vaccine developed in partnership with Arias, which is now IAVI, acquired by them. So the strategy has been that we know that a single company or a single individual or an entity will not be able to carry our agenda forward, the WHO agenda forward. So the best way to do it is to have sustainable and collaborative partnerships, public-private partnerships, but also having a partnership between developed and developing world so that certain discoveries can be passed on and then amplified more optimally in those sites. So proof of concept has been delivered and the rollout now is happening as we speak and is likely to take this objective forward. This was one vaccine, but more are on the way. There are multiple vaccines with different mechanisms or different endpoints that are being studied. And I'm hopeful. I won't be as courageous as my teacher was to say that you and I are going to see the end of tuberculosis, but I'm confident enough in saying that research has come a long way from 100 years back when BCG was developed, and we can now look at many more opportunities to address this problem that we all face. So in summary, we have a problem. It's a global problem. It's a substantial problem with more than 1 million deaths happening every year. We know that the vaccine targets adolescents and adults, and this vaccine performs to a 54% reduction in the incidence of disease. And as a part of a global health strategy, partners will be needed and are in fact being collaborated with to ensure the sustainability of such an initiative. So with that, I come to the end of my presentation, and I would like to thank you all for the patient hearing. And let's all look back in a few years and feel proud that we did our bit to reduce this disease and consign it to history. Thank you very much.